colloquium of 2021, we decided to devote the first three of these events to the various aspects of the current pandemic situation. And so the colloquium today is by Professor Alberto Mantovani, who will be introduced in detail by Antonino Cattaneo shortly. I will just tell you that the next colloquium will take place on March 31st, and the guest in that case will be Professor Rino Rappuoli, who will tell us about antibodies and vaccines. Thank you very much. I will switch off.
So welcome to everyone. It is a real pleasure to introduce to you Professor Alberto Mantovani. It is a pleasure and a great honor to welcome Alberto. He's a most distinguished immunologist and cancer biologist that gave seminal contributions to science. Alberto Mantovani graduated in medicine at the University of Milano, and after specializing in oncology at the University of Pavia, he spent many years first in the UK and then in the US and in other Italian laboratories. And he is currently professor of general pathology at the Humanitas University in Milano, of which he is also the scientific director. He has been the scientific director of the Humanitas Research Institute from 2005 as of today. And he also has an appointment since 2017 as a professor of inflammation and therapeutic innovations at Queen's Mary University in London. Alberto Mantovani is an internationally renowned scientist. He made seminal discoveries. He discovered in the late 70s the pro-tumor function of tumor-associated macrophages, cells of the innate immune system, linking inflammation and cancer. And this discovery puts him as a recognized forerunner, anticipating the times, and the founding father of the inflammation and cancer connection. He discovered a host of chemokines and receptors and their signaling mechanisms, and in particular a new signaling concept of cytokines and chemokines, the decoy receptors, a new concept in biology. In the field of humoral innate immunity, he made a host of a number of seminal findings that gave him the international recognition, for which he was awarded a number of important prizes. I won't mention them all, but these include the International Feltrinelli Prize of the Accademia Nazionale dei Lincei, the American Italian Cancer Foundation Prize of Excellence, the American Association for Cancer Research International Pet Scholar Award for Extraordinary Achievements in Cancer Research, and many, many others. He's a fellow of the Accademia Nazionale dei Lincei, of the Accademia Europea, and of other academies. And he's also very much engaged in public awareness and outreach of science. This is, in addition to research, this is an important commitment that Alberto Mantovani takes, and it is important not only to be an outstanding scientist as he is, but also to fight for the fact that science is communicated to the general public. And he founded the Gruppo 2003 for the advancement of science in Italy. He founded Scienze in Rete, that is a very good, very important outreach blog and journal for the public. So, without further ado, I welcome warmly Alberto Mantovani for his talk that will be on innate immunity and inflammation from cancer to COVID-19. Thank you, Alberto. Thank you for coming.
Thank you, Antonino, for the kind introduction. And I want to thank Augusto Sagnotti for the pleasure to be part of this series of colloquia. And it's a real pleasure to be at Normale. I regret that I'm there only via web in a virtual way, and I hope it's an arrivederci in a physical way. And now it's the most difficult part of my talk, and that's sharing my screen. Can you see my screen?
Not yet.
Oh, gosh.
While you talk, I say something to the public. I forgot saying that those of you who will want to ask questions after Alberto Mantovani's talk can write the question on the chat. I will read them to the speaker at the end of the discussion, or you can raise your hands and I'll try to give you the word. Thank you.
Now, the pointer, and here we are. So again, once more, thank you. It's a real pleasure. In the first part of my chat, I will share with you some fundamental concepts of the logic of the immune system. Then I will dwell a little bit on immunity, inflammation, and cancer, and introduce the general concept of an immune revolution, which has happened in the last 15 years, more or less, 20 years. And then I will move to COVID-19. And I may anticipate things that Rino Rappuoli will discuss, and Rino is he doesn't need any introduction. So this is Thucydides, and it's the Peloponnesian Wars. And as far as I know, this is the first description of immunological memory, because he realizes that the people who survive plague don't get sick a second time or get the disease in a milder form, which is in fact what we do see. I also want to emphasize that there is useful, I think, fantastic contamination between literature and science, and the Piccolo Teatro di Milano. Claudio Longo, he is the current director of Piccolo Teatro, and Daniela Minerva and myself put together this effort, again, mixing literature from Thucydides down to Manzoni and Philip Roth with the immunological comments, and you may want to have a look at that. So the immune system, this is the immunological orchestra. It's extremely complicated. I'm still amazed that we don't know all the players and all the instruments played by the immune system. Another way of looking at the immune system is an army, a hierarchical army. It's an army which is in continuous war. So we estimate that there are a few 100 million of cells of the first line of defense die each and every day to sustain equilibrium with a very complex immunological, microbiological world. So the immune system is, together with the central nervous system, is the most complicated system in our body. Similar investment in terms of weight, number of cells, even more. Connections are key to the function of the central nervous system as well as the immune system. We call them synapses. They're obviously very different. These synapses are transient. We don't really have an estimate for numbers, but again, the principle is that communication is key. And in terms of general keywords, there are common keywords, including recognition. I will get back to the issue of recognition of self versus non-self, recognition of microbes, the good guys versus the bad guys. And we really don't fully understand how the immune system deconvolutes the information of microbes and distinguishes between the good guys, who are essential for our health, and the bad guys. So recognition, communication, of course, and memory. And of course, memory is the basis for vaccines. And I should say that there has been a change of paradigm in terms of memory, and we still don't really understand how memory works. I mean, when we tell you that we don't know how long will last memory to COVID-19, to the vaccines, that illustrates how ignorant we are. So the immune system is made of two compartments. The first compartment is innate, what we call innate immunity, the first line of resistance, originally discovered by Mechnikov. By the way, the seminal experiment was done in our country, in Messina. And he realized that phagocytes, the cells that eat, play an essential role in recognition and resistance. And actually, can you please switch off microphones? And actually, we estimate that, first of all, the innate immune system deals with more than 90% of the encounters we have every day with bad guys. And I will mention some data concerning the importance of the innate immune system in resistance to COVID-19. In addition, more than 90% of living organisms on the planet live with just this line of resistance. This movie, which with the pointer doesn't work, but anyway, this movie doesn't go back to Mechnikov, but it's West Nile virus. And in this particular case, macrophages, the phagocytes, serve as Trojan horse. So there is a yin-yang in the role of the innate immune system, although it plays a key role in defense. About 300 million years ago, there was an explosion in terms of diversity and complexity, and the adaptive immune system was born. The one that is responsible for rejection of grafts. And this is the two saints, Santi Cosma and Abbiano. This is Beato Angelico. And this is a miracle. And I should say that medicine and immunology are doing miracles each and every day, doing successful transplantations. And the general principle of the adaptive immune system is that of recombination, mixing of DNA segments, and the mix that work in terms of production of antibodies or recognition structures used by the directors of the immune system, we call them T cells. The rearrangements that are productive, we call them productive in our jargon, are selected in a Darwinian way. And this is the repertoire of the orchestra in terms of adaptive immune system, the ensemble of recognition structures. So this is one of the founding fathers of medicine, Paul Erlich. And he and people like Mechnikov and more, I mean, were literally seeing a major change of life of humankind on Earth, namely life expectancy moving from 40 years, and it has been like that for 200,000 years for every newborn of our species. Life expectancy moving from 40 years to 80 years. And these are pioneers in immunology. He's depicted, these are antibodies. I could use this representation of antibodies with my students. And these people, and of course they were seeing vaccines, changing life on Earth. They were seeing antibodies, taking care, for instance, of diphtheria of Krupp. And they were dreaming of using immunological approaches to treat cancer. Of course, time was not ripe. And this is a schematic representation of the long pathway of tumor immunology and immunotherapy. And it has been a long pathway. Here is the innate side of the path, long, as was mentioned by Antonino, thank you, I have long been connected with this part of the pathway since the late 70s. And I want to emphasize that along these pathways, we have had many failures. And actually, at the turn of the millennium, this is a Japanese painter, Hokusai, and I don't know if you can see, I mean, we were rowing against the current or in the waves, and tumor immunology was not getting much credit in terms of chances of really changing the natural history of human tumors. This is, please just look, this is more specialist, but if you just look to the right side of this cartoon, at the turn of the century, two colleagues, Doug Hannan and Bob Weinberg, in a paper in Cell, made an effort to crystallize all we knew about cancer, and this is how the message that came out. 6 hallmarks, and all of them have to do with tumor cells, with the cancer cells, with transformed cells. Of course, I didn't agree with this this view, and immunologists at large did not agree with that view, because we felt that the tumor microenvironment, the ecological niche that surrounds cancer cells, we felt that the ecological niche, the microenvironment, is as important as the tumor cell itself. And I proposed actually that there be a 7th hallmark of cancer, namely cancer-related inflammation, as represented by macrophages, which I have studied throughout my life. And this is science. Paradigms change. This is the new version of the paradigm, which includes tumor-promoting inflammation and adaptive immune responses, and these are tamed, suppressed adaptive immune responses as we see them in our patients. I should say that I really don't agree, 100% agree with this. I think that these two should be close together. So in the first 10 years of the new millennium, there has been a paradigm shift with the acceptance of the microenvironment as a key determinant of carcinogenesis, and at the same time, the identification of molecules which represent brakes for the immune system. The brakes of the immune system, which are activated here, we call them checkpoints, and targeting 2 checkpoints has changed the time course and the prognosis of a number of human tumors. And this is a case in point. I'm just using this as an example. The case in point is melanoma, no advanced melanoma, no cures with conventional chemotherapy, first generation checkpoint blockade immunotherapy, about 20% cures, and now about with 2 monoclonal antibodies targeting 2 checkpoints, the estimate is around 40% cures. And here is the challenge. In other words, the 60% that we don't, or the 50% that we do not cure. But let me emphasize a few general aspects. Why don't we cure cancer with surgery or radiotherapy or conventional chemotherapy or targeted therapies? Therapies which are targeted to the genetic events that cause cancer. Because cancer is a moving target. Tumor cells are characterized by genetic instability. But from an immunological point of view, genetic instability represents an Achilles heel, because genetic instability means new antigen, and if you unleash innate and adaptive immunity, you may have a chance to develop effective therapy. Then memory, I will get back to memory. I mentioned repertoire. There is a very large repertoire of recognition structure in the immune system. It's not infinite, but very, very large. And then the potential. This is one of the challenges that we have, namely combine in a rational, effective way immunotherapy with conventional therapy such as chemotherapy or targeted therapy. So we have entered a new word. There is a definition of immunorevolution, and we have challenges ahead of us, and I just illustrate this gap here as an exemplification. And this is the most successful tumor that is treated with immunotherapy. The challenge is the patients that don't respond, the few tumors in which there is no benefit whatsoever in any patient, and the use of other forms of immunotherapy. In particular, for instance, since I'm interested in innate immune cells, myeloid checkpoint immunotherapy. So this is my traditional area of interest, in addition to the discovery of molecules and paradigms, fundamental research in immunology. And almost exactly 1 year ago, we were confronted with a new enemy, a killer, that's SARS-CoV-2. And this is the conventional representation of the virus. This is the virus as seen by 3 of my 8 grandchildren. And in a way, it is as accurate as a representation as this one, because this is, of course, you can see the spike protein, the anchor that is used by the virus to enter the organism. And confronted with a new challenge, this is how I felt. I realized, and I still realize, that I don't know. And this is the premise to develop. This is the challenge. I don't know, then this is the premise to understand something and contribute to understanding. The second challenge here is to share knowledge. And this is an effort that the Accademia dei Lincei has done, issuing a periodic report, a report on vaccines. And if you are interested in fundamental, well, it's very difficult to keep abreast with a fast pace of knowledge, but if you are interested in having a sense of fundamental mechanism and tools in vaccinology, I direct you to the Accademia dei Lincei website. So this is a schematic representation of the interaction of the virus with the organism and with components of the immune system. And I'm going to go through this pathway and emphasizing a few selected aspects. The virus, well, there's no need to comment about, I mean, the things that were said in last spring, during the summer, of a spontaneous attenuation of the virus, which had no counterpart in the scientific literature whatsoever, have been dispelled by the epidemics. And no need to comment about
the page disappeared.
So this is, again, as I said, the schematic representation of the virus's interaction with the immune system. Of course, the receptors. I should emphasize that the virus enters the central nervous system. I mentioned the central nervous system, and the receptor is different. It's neuropilin-1. The interaction of the virus with the host depends on the viral load. We had a break, and hopefully we will have a break in springtime and in the summer, because less viral load. Aging, we all know, and there is a general paradigm of aging which is related to the inflammatory tone, inflammaging. Lifestyle, obesity, for instance, and obesity again impacts on immunity and the tone, the inflammatory tone. And then genetics. And of course, all of us, I mean, have this question, why one guy gets sick and the other one doesn't? Early on, we asked the question whether there is genetics involved, for instance, genetics in the apparent susceptibility of northern Italy. And then we were part of a consortium investigating genetics. And I'm showing this slide only to emphasize a general aspect. And this is the effort of a European consortium. We identified genes on chromosome 3, and genes, some of them are dear to my heart because they are related to chemokines, which we provided one of the very, very first descriptions of chemokines. So molecules related to innate immunity and inflammation. And here is in this Manhattan plot, there is a peak, blood groups. Now this part is rock solid because it has been confirmed, also recently a paper in Nature, and extended, of course. This here is a question mark as to the actual relevance. So this is just for the people who are not involved in science. This is science. I mean, you get data and then you are humble. And if a piece of data is not confirmed, there is a question mark here as to the actual relevance of this. In addition, so genetics. Then the first line of resistance, I will get back to that. The directors of the immunological orchestra, uncontrolled inflammation, a new disease called MIS-C, which affects pediatric age, adolescence, I would say, after infection with the virus. Again, an inflammatory disease. We don't understand why. And then for a long time, this is the only tool that we have that has been proven in a controlled clinical trial or trials and has been shown to reduce mortality. The old cortisone, glucocorticoids, which have been shown by Horby et al. And has been confirmed. And this is, again, we want our data be confirmed. This is a colleague of mine, and this is the general guideline by WHO, which stemmed from these clinical trials. I should say that we are concerned. We are concerned because the evidence relating decreased mortality associated with the usage of dexamethasone or other glucocorticoids only applies to a restricted phase in the natural history of the disease. And we are very concerned by the fact that in our hospital and in general, we see patients being treated early in the disease when we do not want to have suppression of the immune system. And this is, we just published a short paper on this. Let me go back to the first line of resistance, because I would like to share with you the general feeling I have, or view I have, that the perception that we have of the disease has changed. This is vaccine. I will skip this graph to the, and this is why science is useful and is useful in a week. So 3 weeks ago, we reported that patients who had experienced symptomatic COVID upon injection of the first dose of vaccine have a fantastic immune response. And so we feel that there is no need of a second jab. This has implications at the time of vaccine scarcity. A similar report has been published by Florian Kramer in US. There are 3 more reports. The Haute Agence de la Santé has recommended the French government to postpone the second jab. and we may save 1 million second jabs, up to 1 million in our country, if there is agreement that this is the way we should do. So we will wait for decisions. Again, innate immunity, work done at NIH by Gigi Notarangelo and Jean-Laurent Casanova, both, I would define them molecular pediatricians, discovering that a subset of patients have what we call now inborn error, immunodeficient, genetic immunodeficiencies, which is unmasked by the virus. In addition, patients have autoantibodies, so the virus plays tricks, and our immune system in a subset of patients generates an autoimmune response. And in addition, we and many others have identified new markers for inflammation, which are candidate new markers that help physicians. So I want to emphasize the general concept at the bottom of this line, namely that our view of the disease has changed in the last year. It is now, we see them, or I see it, at the intersection of genetic predisposition, chromosome 3. Actually, I should emphasize that it's related to our crossbreeding with Neanderthal, probably, the genes that are relevant here. Actual genetic immunodeficiency unmasked by the virus, autoimmunity triggered by the virus, and uncontrolled inflammation. In the last few minutes, I want to discuss with you viruses. I think it's inevitable, and this is Jenner, the physician who changed life on the planet. Let me also emphasize that there is nothing new in Novax movements. We heard about concern about genetic changes induced by vaccine, for instance, by mRNA vaccines. Nothing new. This is genetic changes induced by vaccination with the vaccine that, of course, they come from cows, from bovine. So there has been a vaccine race. About 1 year ago, there were about 300 candidate vaccines. Now they are down to about 100. There are about 40 vaccines in clinical trials, and 3 vaccines approved in our countries. Possibly we may go up to 5, 6 vaccines approved. Again, this is an effort done as Accademia dei Lincei, Guido Forni, and a group of us, a bunch of us. The prime target for vaccines is the spike protein. And I want to emphasize that had not fundamental work been done on a similar protein of a respirator, at NIH and others, I mean, we would not have a strategy to target this in vaccines. So again, once more, one should give credit to fundamental research. There are various platforms, traditional platforms, inactivated virus, for instance, the Sinovac vaccine, inactivated virus, more conventional protein plus adjuvant. Rino will discuss this at length. He has been a pioneer in the general field of vaccines. Then we have the mRNA vaccine. Let me have 2 comments on mRNA vaccine. The first comment relates to the Moderna vaccine. Without the fundamental research done at NIH, no Moderna vaccine. The BioNTech Pfizer vaccine, I happen to be with the BioNTech people. I mean, this was a strategy that was meant to generate vaccines against cancer. So we go back to the first part of my talk. and Chris Huber, the mentor, and Ugur Sahin. So it's a gift of anti-cancer research. And then I want to comment on this is the virus vaccine, and the various virus platforms have been tested. Failures in the last 10 days, 2 candidate vaccines have been dropped in phase 1. The one that all of us know is the Oxford AstraZeneca vaccine, and here are some of the others. And here again, I want to emphasize 2 aspects. First of all, fundamental research, the Oxford group. The second one is the failure. I mean, these people had generated a vaccine that was meant for SARS, the first SARS. Apparently, it didn't help, was no need, but it provided the basis for developing all these vaccines. Is it good to have many vaccines? Is it good to have different vaccine platforms? Yes, I have no doubt, for many reasons. And one of the reasons is the experience with polio. We live in a continent that is polio-free. In August, WHO announced that Africa is polio-free. Most of the world is wild-type polio-free. This extraordinary achievement has been reached thanks to 2 vaccines which have been used in a complementary way. And we have a dream of living in a world polio-free, and for that we need 2 vaccines plus another one, and then we will stop vaccinating against polio. More than 1 vaccine platform is extremely, or can be extremely useful. Second general message, going back to the innate immunity, that's which of course is dear to my heart. And this is an essay that Mihai Netea and I wrote because I did some of the early prehistoric work. Some vaccines have the capacity to train the first line of defense. We call them trained innate immunity. This term was coined by Mihai, and he worked out the molecular mechanism, epigenetic modifications. And of course, we don't know whether the vaccines are using trained innate immunity. Trained innate immunity underlies a phenomenon that is being called pathogen agnostic protection. What do we mean by pathogen agnostic protection? We mean that you get a shot for measles and you are protected against several infectious agents, respiratory infection. Of course, not to the same extent as measles. We don't know whether the current COVID-19 vaccines are a good training set for the first line of immunity, but in general, the message I would like to leave you with is that the fitness center for immune system are vaccines and lifestyle, as well as a clean environment. Finally, a look at global health. I have served for 5 years in the Global Alliance for Vaccines and Immunization. It's called Gavi. So Gavi is not only a good Italian wine, it is also a global health initiative which has improved health worldwide, in particular in the poorest countries in the world. Let me remind you that at the turn of the millennium, it is estimated that 2.5 million children died every year because they did not have access to the most elementary vaccines. Now the figure is down to 1.5 million, all too many. So Gavi has been the main driver of a global health initiative called COVAX, and the objective of COVAX is to provide vaccines to the poorest and in particular COVID-19 vaccines to the poorest countries in the world. And I think we should never forget that 1 out of 5 child born on Earth does not have access to the basic vaccines. Again, Rino is going to give a talk, and it's a pleasure to quote Rino Rappuoli. Before the COVID-19 pandemic, Rino, Angela Santoni, the president of the Italian Society for Immunology, and myself had a paper in Journal of Experimental Medicine, and the title was that vaccines are an achievement of civilization, a human right for each child born on Earth, and a health insurance, or if you pass me the definition, a safety belt for the future. And what has happened, I think, testifies the truth in this statement. I would like also to emphasize that equitable access to COVID-19 vaccines is not only an ethical duty, which some of us may feel strongly, it is also a matter of safety. We should not forget that 2 of the 4 variants that are a matter of concern for us and for vaccines and for the immune system were born in Brazil, in the Amazon, and in South Africa. So back to our roots in Greek culture, COVID-19 has reminded us that nature is fond of hiding herself. And I think that the challenge is, these are the cuts in this painting by Lucio Fontana. I think that the challenge is to go beyond the cuts. And give talks to high school children, and before the COVID-19 epidemic, I gave a talk on cancer immunology, immunotherapy. And this painting is not, she's not a famous, but she's a teenager. Alice Coppini had this one, and I would like to think that in general it means immunology and vaccinology embracing global health with equitable access to vaccines. So again, once more, it's a real pleasure and a privilege to be at Normale, and thank you.
Thank you very, very much, Professor Mantovani, for your very, very clear and deep talk, which is now open to discussion. And I welcome the audience to either raise the hands or to type a question. And whilst I wait, here there is a raised hand. Augusto Sagnotti, go ahead, Augusto.
A curiosity, at some point you were talking about the success of only a single dose of vaccination, but you did not mention which vaccine you were referring to.
Yeah.
I don't know if it's relevant for the discussion, but it's a curiosity. So.
Yeah, well, first of all, let me qualify the statement. Single dose vaccination only in subjects who have experienced symptomatic COVID.
Okay.
Unfortunately, there has been a call for single dose vaccination independently of, can I make a more general comment?
Please.
What do we know about single-dose vaccination? Well, first of all, we have 2 platforms at present. The mRNA platform is revolutionary in terms of technological strategy, but it's conceived for 2 doses, a prime and then a boost. And we know that the first dose confers some protection. It is partial, and it is observed at 20 days, so it's transient. And the British government decided to took a risk and decided to postpone the second injection also with this type of vaccines because of the desperate situation in which they were until a couple of weeks ago, 3 weeks ago. So it's understandable, but it's not supported by most people. There is the adenovirus. All the adenovirus were born as single-dose vaccines. So the Oxford vaccine, Oxford AstraZeneca, was born as a single dose, and then in terms of based on longevity of the response, they moved it at 2 doses. It's a different game. What we have, the study we have done is 150. I think it's interesting in general because it gives you a feeling of how we do science. We were vaccinating the healthcare workers in our institution. We have a program of monitoring and research. And after the first 150, we realized that the ones who had COVID-19, symptomatic COVID-19, had a tremendous response. The same observation was made in US by Florian Kramer, and last Saturday, 3 more papers appeared. So we put the paper immediately in open access. As soon as we were confident about the data, we had the data in open access. I understand that our data and the data Florian Kramer provided the basis for a suggestion in France. Today I received the message that the same suggestion is being made in Belgium, and we have shared our data with the Ministry of Health and IFA. So we'll see what they decide. But we think that we could save, first of all, we could save side effects to subjects because the vaccination has side effects, and we could save vaccine doses at a time when we're running low in terms of vaccine doses.
Thank you. This question and this answer touched many of the hot issues at the moment. But here we have now a student of ours, Luca Fusar-Bassini, who brings us back to evolution and with an interesting question. I think that probably, Luca, the best thing is that you wrote your question, thank you, but probably you may want to ask the question, which is an interesting one. Luca?
Sure. Well, thank you for the talk. My question is maybe a bit naive, and it does not strictly relate to today's talk. I wonder, why don't eukaryotes use some RNA-based system for specific recognition in immunity? Because I know that we have toll-like receptors, but their specificity is mild to my knowledge, whereas in bacteria, CRISPR systems are relatively ubiquitous and their specificity is advantageous for bacteria. So I imagine that eukaryotes could have separated from prokaryotes before developing CRISPR systems, but I still wonder if maybe there is some less known RNA-based recognition system in eukaryotes that could work in parallel to antibodies versus intracellular pathogens. So thank you, and thanks again for the great talk.
Yeah, thank you for the question. I don't have, well, first of all, nucleic acid recognition systems are complex. And it's interesting because you have, and if you think it over, I mean, they are strategically located. You have receptors, innate immunity receptors that are located on the cell membrane, the toll-like receptors that you mentioned. Then you have sensors of innate immunity that are located in the endosomal compartment, a subfamily of toll-like receptors. And then you have a large complex family of cytoplasmic receptor, which I think addresses some of your point, at least in part, your point. And these are nucleic acid receptor sensors, the inflammasome and complex structures like this. And if I can elaborate, can I elaborate, Antonino?
Of course, we want you to elaborate.
I mean, if you were in presence, I would have asked the students a question. In my scheme, is there anything, an important point that is missing? I mean, you cannot answer, of course, but so I can provide you the answer. The answer is that, you see, we know that innate immunity is important for resistance and pathology. We know that the interferon pathway in innate immunity is essential, and the genetics, the genetic associations, and the bona fide immunodeficiency unmasked by the virus tells us that it's essential. We know that there have been case reports of a couple of genetic deficiency, one of the receptors that I mentioned, but it's amazing. There is a missing link. We don't know what the immune system recognizes of the virus. We have a story submitted for publication, but I cannot comment on that. But it's a very, very interesting question.
Is there another question? Whilst we wait for it related to this, I will ask one. So you mentioned that the big explosion in 300 million years ago in the shift between evolutionary shift from innate to adaptive responses in the immune system. And of course, that discontinuity also occurs in the change in the logic of how the nervous system is organized. Because our nervous system is organized in large numbers, statistically organized cells, and not all connections are pre-specified, whereas in vertebrate, before the discontinuity, each connection is pre-wired. And I wonder if there is something in common in your mind in this big evolutionary leap between invertebrates and vertebrates. Of course, in the immune system, we know that recombination is one important. Yeah. So I wonder if you can comment on this.
Well, I have two comments, and one is one comment relates a general comment, something that I didn't say, namely those are the due massimi sistemi. Due massimi sistemi, I think. I mean, I think the data suggests that those are the due massimi sistemi. We know that there is a dialogue between the two, but we really understand very little. I mean, there is clinical phenomenology, for instance. I mean, we know that, I was educated as a medical doctor, and I live in a hospital. We know that, for instance, some patients with ulcerative colitis get very bad under conditions of stress. We know that in subjects with asthma, there is some kind of connection. And we start to understand the molecular mechanism and the pathway. But I think that, that general field of understanding, and I didn't know that in evolution there was, at about the same time, major change also in the central nervous system. I think that is an absolutely fascinating area. Michela Matteoli in our place is working on the connections, and I think it's a most fascinating. The second general point that I want to draw your attention in terms of evolution of the adaptive immune system. And you have rearrangement of gene segments that generate antibodies and the T-cell receptor and the diversity and the repertoire. And I learned from a friend of mine, Max Cooper. By the way, he's a clinician and pediatrician. And Max discovered that there is a major division in the adaptive immune system in agnatostome, without jaw, and agnatostome. And I'm amazed that here are Ig domains that are recombined, whereas there are leucine domain. So the tools are different, but the logic is the same. I'm fascinated by that. And perhaps one day we will have monoclonal antibodies from lamprey, and they will help us against future challenges. Fundamental research.
Lamprey bodies, beautiful.
Lamprey bodies.
Other questions from the floor, as we say, as we would say if we were in a room. I don't see any typed question. Okay. If I may take the privilege of being the chair, I'll have another question, more clinically oriented. You mentioned the temporal windows in the dexamethasone treatment, and this relates more generally. I mean, the whole issue of temporal windows is, as I read, also important for monoclonal antibodies that are being considered. And so this is very much related to a general issue with inflammation. There is a positive inflammation and negative inflammation. Do these temporal windows relate also to the different phases of inflammation?
I think that this is the case in a schematic way. Early on, you want to have the immune system, and inflammation is a manifestation of innate immunity. And early on, it's conceivable that you want to have your immune system as a whole to run as much and as fast as possible. And in the later phases of the disease, the inflammation is out of control, and you have a systemic inflammatory response syndrome leading to multi-organ failure. And that is probably the time when you want to use brakes. And dexamethasone is a low-cost brake. And I want to emphasize that when Horby is the guy in UK, proposed, I understand, to the British consortium to run a trial. I mean, he ran against current wisdom of the time, because my colleagues who worked on conventional acute respiratory distress syndrome, there was no evidence clear evidence for activity. There was an official recommendation by WHO not to use glucocorticoids. So, I mean, there is a value in running and in swimming against the current. And there is now and again the value of failure. Let me emphasize the value. I mean, if there are young people who listen to us, all clinical trials blocking cytokines through a month ago, they all failed. You all heard about tocilizumab and people going on TV and saying this is the cure. Nothing worked in a clinical trial. And we have to accept that. However, now there is evidence in the recovery trial, again Horby, showing that again in the right window, it can work, which I think is a matter of hope.
So the question really will be markers for beyond the clinical evaluation for these windows, I guess, or how you evaluate these windows by clinically.
I think that we are doing in 1 year what in cancer research has been done in 50 years, namely dissect the diversity of the disease in terms of individuals, genetics, phases of the disease. For that, we need new markers. We have made an effort, and we have a candidate new marker, which is very, very promising. And hopefully that will help the day-to-day management of the patients, as well as the clinical trials, to test I mean, the objective is to give to the right patient, the right diagnostic, and the right therapy.
Thank you very much. And I think at this point we can only give you a big, big applause and thank you really wholeheartedly. And I really hope to be able to welcome you in Pisa nella Sala Azzurra, nella Sala degli Stemmi a Scuola Normale. And I think that Augusto Sagnotti wants to give a last remark. Augusto, please.
Thank you very much. It was very illuminating and very well designed for our audience. Thank you very much.
Pleasure. It's been a pleasure to be with you.
Thank you, grazie mille, grazie.
Arrivederci, ciao.
Arrivederci, ciao, a presto. Arrivederci. Grazie.
Grazie.
Ciao a tutti.
Ci sono molti ringraziamenti nella chat per